Hello, my name is Alex Bean, and today we'll be talking about three things: recombinant DNA technology, the polymerase chain reaction, and gel electrophoresis and DNA fingerprinting. All of these are very closely related to cloning, which refers to any process in which genetically identical copies of an organism or their DNA or their cells are produced as a result. So I drew a little diagram of roughly what it would look like for an embryo to separate in the womb, resulting in what we know as identical twins, which is a natural form of cloning. So for now, let's focus on recombinant DNA technology. Recombinant DNA technology refers to a method of cloning involving the DNA from two or more organisms through the use of these things called plasmids. But what exactly are plasmids? Well, plasmids are these rings of DNA in prokaryotes that exist separately from the main chromosome. And let's talk about the anatomy of a plasmid. So, it's in the shape of a loop, but for the purpose of this explanation, I'll draw it in a circle, and it has two rings to represent the sugar phosphate backbones. So, plasmid vectors are in the shape of a loop, and they're comprised of an origin of replication. This is where DNA polymerase initiates replication, so it's able to replicate independently in a cell. And it also is comprised of an antibiotic-resistant marker gene and its own genetic data for non-coding genes. So here's the gist of it. You have a strand of DNA here containing a gene that you want to be coded, and restriction enzymes like EcoR1 cut out a specified gene along the code GAATC, and it also makes a cut on the plasmid. So this gene is left with ends that do not have attached complements, and these are known as sticky ends. Notice how they are only able to connect. Notice how the gene and the plasmid are able to connect because they use the same restriction enzyme, and that's very important. Alright, so when the genes are pasted into the plasmid, and their complements on the sticky ends are matched up with that of the plasmid, the DNA ligase glues them together in a sense by forming phosphodiester bonds on the backbones between the sticky ends. And now we have a recombinant DNA molecule. So with this complete recombinant DNA molecule, we now want to clone it. How do we do this? Well, a recombinant DNA molecule is often referred to as a vector because it is a medium through which the DNA is introduced to a host cell, and for replication, this vector is introduced into a bacteria such as E. coli because it is an organism that can do the cloning for us. But how do we get it in there in the first place? First, we must acknowledge what keeps the plasmids from entering the bacteria in rates that are sufficient for lab testing, which is the cell membrane. The cell membrane is comprised mainly of phospholipids, which are hydrocarbon tails attached to a phosphate head, which carries a negative charge due to their attached oxygens. So they're comprised of these phospholipids and pores. Now let's take a look at the anatomy of DNA. We will see that the backbone of DNA also carries negative charged phosphate groups, and so, operating by the basic rules of magnetism, the two like negative charges repel each other, making it very difficult for the DNA to get inside the bacterium through these pores. Alright, so we want to get the plasmids into the bacteria. How do we do it? First, we get a calcium chloride solution and put the bacteria and the plasmids into the solution because calcium chloride neutralizes it, having the calcium ions negate the negative charges of those phosphates so they don't repel each other. Then, the second component is called a heat shock. And a heat shock is basically what it sounds like. The solution is brought down to 0 degrees and then raised and then lowered again. The initial cooling is done so that the phospholipid bilayer contracts, making it easier for the calcium ions to shield the phosphates and keep their charges neutral. Then the solution is heated rapidly so that it's on the hot extracellular side of the bacteria, while it is still cold on the intracellular side of the bacteria. Because it's hot on the outside of the bacteria, that is where the molecules move more because there's more energy. And as per Newton's second law of thermodynamics, the plasmids move from the hot exterior of the cell to the cool interior of the cell, or the bacteria, where the plasmids can then be copied via bacterial replication. However, it's important to note that this method does not always work, so we need a way to weed out the bacteria that do not carry the plasmids with the desired attached gene. Remember before when I mentioned those antibiotic markers? This is when they come into play. So in order to weed out the bacteria without the desired plasmids, we apply the bacteria in the solution to a petri dish containing nutrients such as agar gel that are suitable for sustaining life along with an antibiotic which is shown in red in the drawing. And depending on the antibiotic resistant marker that was put into the plasmid, the antibiotic on the petri dish should match it so that the bacteria with the desired plasmids are not killed by the antibiotics. For example, let's say we were using a PUC18 plasmid, which contains an ampicillin-resistant gene and a beta-galactosidase gene, and I'll talk about the beta-galactosidase in a little bit. I would like to apply the antibiotic ampicillin to the agar in order to kill any bacteria that don't contain the gene for resisting the ampicillin antibiotic. Another way to make sure we've got the right bacteria in the case of PUC18 plasmid, we use the beta-galactosidase gene, and this gene contains what we know as a polylinker, which allows for the placement of the desired DNA fragment or gene. Therefore, if the bacteria contains a plasmid without any foreign DNA, it will leave behind a blue-colored compound in the presence of a chemical called XGAL, and if it does contain the desired plasmid, the placement of the gene will divide the beta-galactosidase gene, preventing it from releasing colored compounds in a situation referred to as insertional activation. So, if the resulting colonies of bacteria don't release blue compounds and don't die when exposed to antibiotics that it should be able to resist, then the resulting bacteria contains the desired gene for cloning. So, cloning is cool, but what use is it in the real world? This discovery by Stanley Cohen and Herbert Boyer is revolutionary in that it confirmed the genetic material could be transferred between species, as is evident through their multiple experiments with Staphylococcus and E. coli, as well as with frogs and E. coli. Through this, they hypothesized that the same could be done with genetic data from humans, eventually leading to the establishment of the biotechnology industry, where they successfully synthesized a growth hormone called somatostatin through the recombinant DNA technology, eventually leading to the cloning of human insulin, which has saved the lives of countless diabetics. And that's what we know about recombinant DNA technology. So, we've talked about recombinant DNA technology. Now let's talk about the polymerase chain reaction. The polymerase chain reaction is an alternative to recombinant DNA technology in that it eliminates the need for a vector, and it results in much more cloned DNA in a shorter period of time. It amplifies the desired DNA sequence by mimicking DNA replication in an actual cell using only heat-resistant DNA polymerase, primers, nucleotides, and of course the desired strand. But how exactly does it do this? The first step in PCR is to denature the DNA, and by denature we mean separating the double helix into its individual strands, separating the base pair complements, and this is done by heating the solution up to about 95 degrees Celsius for 15 seconds. The second step in the polymerase chain reaction is to hybridize the DNA primers onto the DNA that we want to replicate, and by hybridize we mean attaching the primer to the 3' end of the complementary strand so that the DNA polymerase can attach and code from the 5' to 3' end of the complementary strand. This is done by cooling the solution down to about 54 degrees Celsius, because this is the temperature at which the DNA primers will bind onto the flanking sequence, that is, the sequence located just outside the target sequence. It is these same primers which keep the DNA strands from reforming their hydrogen bonds and reverting back into DNA. The third and final step is DNA synthesis, where the solution is raised to about 72 degrees Celsius, and the heat-resistant DNA polymerase known as Taq binds to the DNA and begins transcription. As you may have noticed, the number of strands of DNA increases exponentially after each split and can be represented by the equation 2 to the n, where n is the number of copies made. So, we are able to literally copy millions of DNA sequences from only one using the polymerase chain reaction. So what? What do we do with this abundance of copied DNA? Well, as we will see in a little bit, it becomes most useful when used in coalition with gel electrophoresis and DNA fingerprinting, where an abundance of DNA is used with restriction enzymes to examine specific DNA fragments and/or use the patterns that said fragments form in agarose gel to identify and compare or differentiate the DNA from two sources. So in gel electrophoresis, we start out with a bunch of DNA fragments containing short tandem repeat sequences with a certain amount of restriction enzymes. And for those of you who don't know, short tandem repeat sequence DNA is a repeating sequence of intronic nucleotides. So, for the purpose of this explanation, we'll be using two restriction enzymes, X and Z. So we have three test tubes of identical DNA in a solution, and one tube of size standard solution. One tube has restriction enzyme X, one tube has restriction enzyme Z. One has both enzymes X and Z. A blue, slightly negatively charged dye is added to all of the test tubes. We also start out with a slab of agarose gel with 4 wells that are labeled according to the solutions in the tubes. This slab of agarose gel is placed into a buffer solution where positive and negative electrodes are hooked up to either end. Alright, so our experiment is set up, now let's perform it. First we drop the solutions corresponding to the labels in the wells, then turn on the electrodes. But what exactly does this do? Well, the electrodes form an electric field which gives electric potential energy to and repels the negative charges found in the oxygens of the sugar phosphate backbones of DNA and the slightly negative blue dye. This electric potential energy is then converted to kinetic energy due to the opposing force of the positive charge and the pulling force of the negative charge on the other end. And it's also converted to kinetic energy due to the porous structure of the agarose gel, so that the DNA fragments are able to move through the gel. Depending on the size of the DNA fragments, the longer they are, the more resistance they will face in passing through the gel, slowing them down. And inversely, the shorter the DNA sequence, the less resistance they will face and the faster they will travel through the gel. This results in a pattern of bands, the number of which is directly related to the number of restriction enzymes used. In this case, because there was one restriction enzyme used for X and Z, there should be only 2 bands. However, there should be 3 bands for the XZ enzyme solution, and about 10 on the size standard, just for a frame of reference. This happens because, for each restriction enzyme, the DNA molecule is split at one point, as is shown in the diagram. The placement of the bands in the gel is representative of the size of the fragment cleaved by the restriction enzyme, directly correlating it with the number of base pairs. Because the smaller fragments move down the agarose gel faster, the bands in the gel toward the bottom, closest to the positive electrode, are therefore smaller and have less base pairs, and the bands in the gel closest to the negative electrode are larger and therefore have more base pairs. In the case of this experiment, or this example, the band closest to the positive electrode in solution X would be the A section, and in solution Z would be section D, while in solution XZ it would be solution A because they are the shortest strands of DNA in their respective solutions. By looking at the position of the blocks relative to the size standard, we can accurately tell what size the DNA fragments are in the base pairs and the total length of the DNA strand by adding the blocks from one solution together. Okay, so we have gel electrophoresis. Now let's move on to DNA fingerprinting. So what exactly is DNA fingerprinting? Well, it simply refers to any technology in which the same strand of DNA is compared with that of others because, much like a fingerprint, one's genetic code is unique to the individual. The method of DNA fingerprinting used in coalition with gel electrophoresis is done by taking the agarose gel from the gel electrophoresis experiment and soaking it in a basic solution so that the DNA is denatured into its two complementary strands. The gel is then transferred into a salt solution where a nylon filter is placed on top of the agarose block and absorbent towels are placed on top of that, forming the experimental body known as a blot. Operating by the basic principles of osmosis, the water in the gel moves across the semi-permeable nylon filter into the towels, dragging the denatured DNA to the filter because it cannot diffuse through it. This filter is then taken off the agarose block and treated so that the denatured DNA is permanently adhered to the filter. This filter is then put into a solution with radioactive probe consisting of the complementary DNA strands of the DNA sequence we are looking to compare. These complementary DNA strands hybridize with the repeating sequence of interest that are permanently adhered to the nylon filter, and any unhybridized DNA is washed off. X-ray film is then placed on this filter, and the radioactive DNA exposes the silver halide crystals in the film, causing them to clump together into black spots, and these tell us where the complements to the DNA in the radioactive probe are, and therefore the location of the targeted DNA sequence. The band that matches this DNA sequence can then be cut out for further analysis through other processes like high-resolution gel electrophoresis, and this is a process in which matching DNA sequences are fluorescently labeled and then put under a fluorescence detector to determine the amount of DNA in each fragment, and this forms a pattern in a graph that measures size in relation to the degree of fluorescence. Alright, so we've covered the polymerase chain reaction, gel electrophoresis, and DNA fingerprinting, and their importance in everyday life is seen through their contributions, not only to the world of science, but to the judicial system through their implementation, through forensics involving murder cases as well as paternity testing, and it has even more applications in the medical world, making the analyzation of genes for certain illnesses and their heredity possible, as well as the establishment of possible evolutionary relationships. And so its application to the real world is seemingly limitless. So that is what we know about the polymerase chain reaction, gel electrophoresis, and DNA fingerprinting. And so I hope you learned something.